Welcome dear students. Today we will be discussing about the structure of proteins. Proteins are high molecular weight chemical compounds that carry out virtually all of a cell's activities. They are the molecular tools and machines that make things happen. As enzymes, proteins immensely accelerate the rate of metabolic reactions. As structural cables, proteins provide mechanical support both within the cell and outside their perimeters. As hormones, growth factors and gene activators, they perform a wide variety of regulatory functions. As membrane receptors and transporters, proteins determine what a cell reacts to and what type of substances constitute the machinery for biological movements. Such a diverse display of functions performed by a single molecule can be explained by the presence of virtually unlimited shapes that proteins as a group can assume. In other words, proteins are capable of such wide variety of activities because they can exhibit such a great variety of structures. Each protein, however, has a unique and highly ordered structure that enables it to carry out a particular function. The fundamental principle of protein chemistry is that one protein differs from another because each of its polypeptide chains consists of amino acids polymerized in a particular sequence. While physicists study three-dimensional structure of macromolecules, the biologists study protein structure up to 4 levels of organization. These levels are primary structure, secondary structure, tertiary structure, and quaternary structure. Now let's analyze the primary structure. The primary structure of a protein is the specific linear sequence of amino acids that constitute the chain. Each protein has a unique sequence of amino acids that gives the molecule its unique properties. Many of the capabilities of a protein are determined by the chemical properties of its constituent amino acids. There are 20 different types of amino acids that are commonly used in the construction of proteins, whether from a plant, virus, or an animal. All amino acids have a carboxyl group and an amino group, which are separated from each other by a single carbon atom, the alpha carbon. In a neutral aqueous solution, the alpha carboxyl group loses its proton and exists in a negatively charged state, and the alpha amino group accepts a proton and exists in a positively charged state. Amino acids like sugars have asymmetric centers. With the exception of glycine, the alpha carbon of amino acids binds to 4 different groups so that each amino acid can exist in either a dextro or a levo form. Amino acids usually in the synthesis of a protein on a ribosome are always L-amino acids. Microorganisms, however, use D-amino acids in the synthesis of certain small peptides, including those of their cell wall and several antibiotics. Amino acids are classified on the nature of their side chains into 4 categories as polar and charged amino acids, polar and uncharged amino acids, non-polar amino acids, and those with unique properties. During the process of protein synthesis, each amino acid becomes joined to 2 other amino acids, forming a long, continuous, unbranched polymer called a polypeptide chain. The amino acids that make up a polypeptide chain are joined by a peptide bond that result from the linkage of the carboxyl group of one amino acid to the amino group of its neighbor with the elimination of a molecule of water. The average polypeptide chain contains about 450 amino acids. The longest known polypeptide found in the muscle protein titin contains nearly 27,000 amino acid residues. Once incorporated into a polypeptide chain, amino acids are termed residues. The residues on one end of the chain, that is the N terminus, contains an amino acid with a free alpha amino group, whereas the residues at the opposite end, that is the carboxyl terminus, has a free alpha carboxyl group. The backbone or main chain of the amino acids is common to all of the amino acids. The side chain or R group bound to the alpha carbon is highly variable among the 20 amino acids or building blocks of proteins. And it is this variability that ultimately gives proteins their diverse structures and activities. If the various amino acids side chains are considered together, they exhibit a large variety of structural features ranging from fully charged to hydrophobic, and they can participate in a wide variety of covalent and non-covalent bonds. For example, the side chains of the active sites of enzymes can facilitate many different organic reactions. The varied characteristics of the side chain of the amino acids are important in both intramolecular interactions, which determine the structure and activity of the molecule, and intermolecular interactions, which determine the relationship of a polypeptide with other molecules, including other polypeptides. Now we come to secondary structure of proteins. A polymer's secondary structure is defined as the local conformation of its backbone. For proteins, this has come to mean the specification of regular polypeptide backbone folding patterns like helices, pleated sheets, and turns, etc. If we examine the geometry of the protein backbone, it reveals several important features. First, the peptide bond between the two amino acids is essentially a planar. Thus, for a pair of amino acids linked by a peptide bond, 6 atoms lie in the same plane. The nature of the chemical bonding within a peptide explains these geometric preferences. The peptide bond has considerable double bond character, which prevents rotation about this bond. The inability of this bond to rotate constrains the conformations of the peptide backbone and accounts for the bond's planarity. 2 conformations are possible for a planar peptide bond. In the trans conformation, the 2 alpha carbon atoms are on opposite side of the peptide bond, while is in the cis conformation, these groups are on the same side of the peptide bond. Almost all peptide bonds in proteins are trans. In contrast with peptide bond, the bonds between the amino group and the alpha carbon atom and between the alpha carbon atom and the carboxyl group are single bonds. The 2 adjacent rigid peptide units may rotate about these bonds, taking on various orientations. This freedom of rotation about 2 bonds of each amino acid allows proteins to fold in many different ways. The angle of rotation about the bond between the nitrogen and the alpha carbon atoms is called phi, and the angle of rotation about the bond between the alpha carbon and the carbonyl carbon atom is called psi bond. The phi and psi angles determine the path of the polypeptide chain. Despite being free to rotate, not all combinations of phi and psi are possible. G.N. Ramachandran in 1963 recognized that many combinations are forbidden because of steric collisions between atoms. The allowed values can be visualized on a two-dimensional plot called a Ramachandran diagram. 3/4 of the possible phi and psi combinations are excluded simply by local steric clashes. Steric exclusion, the fact that two atoms cannot be in the same place at the same time, can be a powerful organizing principle. The rigidity of the peptide unit and the restricted set of allowed phi and psi angles limit the number of structures accessible to the unfolded form, sufficiently allow protein folding to occur. The early studies on protein secondary structure were carried out by Lewis Pauling and Robert Corey of the California Institute of Technology in 1951. By studying the structure of simple peptides consisting of a few amino acids linked together, Pauling and Corey concluded that polypeptide chains exist in preferred conformations that provide the maximum possible number of hydrogen bonds.